All right, that works. Okay. So thanks for coming. Thanks for not leaving during Jack's jokes. What I want to talk about is a field guide, but I have a little minor interlude here, which is that my slides are posted to SlideShare already. You can look at them there. Post anything you want about this talk. Blog about it, tweet about it, discuss it, share it, mock it. Send bad notes to Jack. Just don't keep it secret. Don't follow these notions that we're not supposed to talk about things. So what I want to start out with is not talking about microbes. I want to tell you sort of a parable that I sort of witnessed from afar as it was happening. And it's about a bird. One of those, what do you call them? Animals, I think? They're blobs. So at this nice bucolic setting somewhere in the world, some people observed this bird, kind of a cool looking bird. If anybody know what it is? So if you weren't sure, you might whip out your field guide to the birds, look it up. You can find, flip through the book, try and find the particular page for this organism of interest. If you're a total dork like me, you have a field guide on your iPhone. So this is iBird, which is incredible. It has an entire field guide. It has songs. It has means to recognize the birds. It's really quite incredible. And so you could also look it up this way with key information, etc. And when this happened, this wouldn't be that interesting a story if it was here. But it wasn't here, it was there. And can you tell where there is? Anybody yet? This is for you, Jack.
Oh, right.
In Grimsby, near that channel that separates that island from Europe. That's why you're not European, right? That's a little unusual because this is an American robin, and it's not supposed to be in Grimsby or anywhere nearby. And all of a sudden, because birders are very communicative, you could say, or total losers, you could also say, they like to share when they see an unusual bird. With social networking, they can share even better than they used to share. So I think they're called Twitchers in England. So Twitchers now use Twitter to communicate about tweeting birds or something to this effect. And so people sent around a message that they saw an American robin, which did not belong in England. Very unusual. All sorts of people came to see this. Unfortunately, they also watched it get eaten by a predator. So they got to witness the disappearance of the American robin from England, as well as the appearance of the American robin in England. Now, I'm not here to tell you about the fun of watching predatory birds catch things, because I actually like all the falcons and hawks and other birds. What I want to talk about is this parable in the light of microbes. So what's amazing about this story to me is how quickly people were able to identify that something was anomalous. This bird showed up in England. There are people who already know the field guide to birds in their head. They have a collection of the bird guides. They have tools to leverage it, like binoculars. I mean, they probably didn't need it for the robin in the yard, but they have tools to leverage that field guide, and they can immediately look up in the field guide where things are supposed to be, how to identify them, what their behavior even is, and whether or not things are anomalous. And as Rob already had, because Rob's a better graphics editor, or has someone who's a better graphics editor than I am, his looks a little better than this, but I dream of having the equivalent thing for microbes, a field guide to microbes, and we really don't have that. And what I want to talk about for the rest of the talk is sort of a way we can get to that point where I think we have a useful field guide. So I want to go into a tiny bit more detail on what field guides are. So a field guide is not just a picture book, right? It is a representation of an enormous amount of knowledge that has been gathered about a group of objects. Usually organisms. There are field guides to all sorts of other things out there. But in terms of organisms, it's a representation of the knowledge of things like the taxonomic diversity of organisms. Is there a pointer here? The taxonomic diversity, the niches where you find things, the ranges of those organisms, the biological diversity, functional diversity, all sorts of information about those organisms. And the field guide part is really, it's been converted, that knowledge has been translated into a simple representation that hopefully you can carry around with you, and that can be used by a diversity of people, including amateurs, like the birders, who are not experts per se in classifying birds, but can make use of that information and use it in some powerful way. Even professionals use field guides too, because the simple representation, it's a lot easier to carry around a field guide than 700 monographs about birds. So the field guides are very useful to everybody in the context of sort of bird studies. There's this great thing that I found last night while working on my talk from the University of Illinois, from this person Diane Schmidt, which is a field guide to field guides. It is completely incredible. She has a database that she's maintained of different types of field guides, and if you click on each of these links, you can find the collection of field guides. She has reviewed everything in there. She has reviewed personally, it says on her website, which is amazing. So there are all sorts of field guides. There's no microbes on the front of this list, although the large multicellular fungi, we might make honorary microbes occasionally. But there are no microbes on this list. However, just as an aside, I collect field guides, by the way. I have hundreds of them. I can't help myself if I go to a used bookstore and buy old field guides. I love them. Anyway, as an aside, so if you click on miscellaneous, which I did last night, there is a reference to this wonderful book, this incredible a book called The Field Guide to Bacteria by Betsy Dyer. And if you don't have it and you're interested in microbes, you have to get this book. It is, in essence, an attempt, sort of a preliminary attempt at having something akin to a field guide for microorganisms. It has things about, seeing the macrological effects of microbes in various places like Yellowstone Hot Springs, etc. It's really quite an incredible thing. So I'm not saying there aren't attempts at field guides to the microbes that are out there. There are, in fact, a whole collection of other things that you could consider attempts at field guides to microbes in a variety of ways. But as you've already heard, and I'm sure most people here know, the representation that we can make in a field guide to the microbes is limited in part because we haven't known a lot to put into those field guides. So these preliminary attempts are very limited because we don't have the information that we need in order to represent in a field guide what we would like and what would make it like that bird story, be able to go to a field site and know what's wrong with the microbes there or what's interesting about the microbes there. So what I'm going to just go through is sort of the categories of things that I think are needed to really start to have something that is akin to a field guide to the microbes. Now, sort of like the Earth Microbiome Project, this notion of a true field guide to the microbes is ludicrous. I mean, the microbial diversity that is out there we can't really handle it in the way that we can handle 8,000 species of birds, for example. Although, as I think Rick already mentioned, there are only 6,000 to 8,000 species of bacteria that have been formally described. But we all, or most of us, appreciate, and I'll get into this in a little bit, that there are probably many, many more out there. And there are hundreds of thousands, if not billions, of niches out there for microbes, and describing them all in a single field guide, it's ludicrous. I admit that. But the goal, the drive to have something akin to what people do with birds, I think is a worthy organizing principle to try and figure out what we need for gathering this information like people have been doing with the Earth Microbiome Project. So what I'm going to talk about first is this part of a field guide that is a catalog of the types of organisms, as well as the taxonomy and phylogeny of those organisms. And so there was a bit of a controversy a few months ago over something related to this with microbes. So again, as I just mentioned, the we have only about 8,000 or so described species of bacteria and archaea. And those described species, people have gone through a meticulous process to formally characterize and describe those organisms. And if you take that information as though it is all of our knowledge about microbial diversity, you end up with what I would call very strange, if not absurd, inferences about the life on the planet. So there was a paper that came out in PLOS Biology that was a very interesting paper if you focus on the multicellular organisms that are studied in the paper. But they tried to take information about the described organisms on the planet and make inferences about how many there might be of different types of organisms. And based upon information about described bacteria and archaea, they predicted that there were, or was, one species of archaea in the oceans. And that's because people have not isolated and described and characterized a lot of diversity of archaea from the oceans. So if you try and make predictions based upon what was known from the described organisms, you end up with just completely silly predictions based upon that. And so many microbiologists, myself included, got up in arms over this prediction. I said some not so nice things in a New York Times article. But I mean, I think this issue is really important. Describing organisms is very useful. Describing type species and type strains. And as I'll get to in a minute, we focus on type strains in many cases for a lot of the biological work that we do. But it is completely insufficient as a way of characterizing the diversity of microbes on the planet. As we've seen, molecular methods have gone out there and characterized microbial communities using ribosomal RNA PCR or metagenomics or other methods, and we have found an incredible diversity of DNA sequences out there in the environment. People have studied many of the organisms from which those DNA sequences come and have not yet or ever described formally a species that those sequences come from, nor will they. It is inconsistent with much of the methods and the approach that people are using. To me, it would be akin to having to have a sample from every star in the universe in order to try and characterize it. We're just not going to do that for the diversity of microbes on the planet, or we will be hopelessly behind in characterizing them. And of course, a large component of this problem is this issue that you've already heard about, the challenges with culturing microbes. You can't grow everything in the laboratory, and so if you require culturing as part of your description of organisms, that's not, broad enough to cover the diversity of life on the planet. So I'm not going to talk about this more. I have lots of ideas about this in the interest of openness related to this article that Carl Zimmer wrote and a blog post that I wrote. I created an open Mendeley reference collection with papers that have studied the number of bacteria and archaea on the planet. There are about 50 papers in this collection now, and anybody can go look at that collection and look at the science behind studying the diversity of microbes and estimating the number of species. It includes references to Barry Whitman's papers, which are, include calculations about the number of organisms, and I'm trying to keep this updated. But there's lots of complications with this, which I'm not going to talk about, like what is a species, and how that applies for bacteria and archaea in particular with issues like lateral gene transfer. And I'd be happy to talk to people about that separately. But nevertheless, there are certainly more than one species of archaea in the oceans. And we need to incorporate alternative methods and not be so constrained with the history of how people formally describe organisms. So when you do this with molecular data, you've already heard a little bit about this. Basically, you can go collect DNA from a sample, do a ribosomal RNA survey, or a metagenomic survey, or some type of other omic-y survey of some kind. And then what you want to do is do some type of phylogenetic, Rob already mentioned this, classification of the sequences that you get. You want to use, as best as you can, reference data from those described organisms, if that is possible, or from other environmental samples, if that is possible. And then you can divide the sequences up into these OTUs. Which one, how far did you get, Jack? 5? We also have OTU 6, OTU 7, OTU 8. I think there are more. So you divide them up into these species-like entities from the sequence data that you would call OTUs. And again, we can debate exactly what level of cutoff to use for OTUs and what it means, but there's nevertheless an enormous amount of diversity out there in microbial sequences. One alternative way to look at this is to not bother with trying to classify things into OTUs and to try and use the total branch length in the evolutionary tree, something called phylogenetic diversity, as your metric, rather than counting the number of things at the tips of the tree that come up into clusters. And there have been a lot of approaches. This is what Unifrac, Rob Knight's tool, and many other tools out there make use of the actual phylogeny of the sequences and how close they are to each other without needing to really worry about assigning each tip into a particular bundle that you would call an OTU. We've done a lot of work in this area, so in the original Venter-Sargasso C paper, we were very excited to help analyze that data because you could scan through metagenomic data and for the first time really look at phylogenetic diversity of organisms using a gene other than ribosomal RNA genes. So if you scan through metagenomic data, you can do the same type of phylogenetic analysis that we've already heard about and that people have used for 20 years with ribosomal RNA. You can now do that for other marker genes, other genes that you think contain some signal about the phylogenetic diversity of organisms. I've been obsessed with Rec-A for most of my life, so this is the phylogeny of Rec-A, but in that paper we used 5 protein coding genes in addition to ribosomal RNA to scan through the metagenomic data. And one important thing to consider is that many protein coding genes have less variance in copy number between taxa than ribosomal RNA genes, than for ribosomal RNA genes, and therefore they can be better estimators of relative abundance of organisms because they might have less variance in the copy number. We don't have as good a database of these other genes, so placing them in a phylogenetic context is much harder because someone should do a count. How many ribosomal RNA sequences are there now? Like a trillion or some? Not fewer than a billion. And the next most sequenced gene probably has 10,000 or something. So it's still got issues. So we've developed, and I think this is really important, you've already heard a reference to this before, with the amount of sequencing that's happening, we need automated, openly available tools to analyze and process this data. Originally, we developed a tool to do this for protein families called Amphora. It's not the best written software in the history of the planet, but you can run it. And we can scan through metagenomic data and assign these protein coding genes to taxonomic groups, just like you can with ribosomal RNA. As an aside, one interesting thing that we're finding is if you build phylogenetic trees of protein coding genes, every one that we've looked at so far, there are branches in these protein coding genes that are distantly related from the branches that correspond to any known organisms, including bacteria, archaea, and eukaryotes, as well as viral sequences. We don't know what these are, but it is telling us we know very little about the total phylogenetic diversity of entities that are out there in environmental samples. A second component, so you can do this phylogenetic classification, and it is very powerful to do this with metagenomic and ribosomal RNA data, and with the cheapness of sequencing, we're going to have a good sample of the phylogenetic diversity broadly, including even viruses, although as Rick mentioned, it's a little bit harder to do that. But we are going to have that phylogenetic diversity characterized, at least at some level. What you need to layer on top of that is information about biogeography. This is the picture of a map in my bird field guide of where the American robin is supposed to be. We need that for all of these OTUs, species, taxa, whatever you want to say, for microorganisms. And I'm not going to talk about this because that is, in essence, what the EMP is partly about, is to go around and collect global samples from across the planet. And again, this, on the big picture level, this is crazy. But in the detail, we need this information to have any idea about things like ranges of microbes, ubiquity of microbes, endemism of microbes, etc. So we have to start somewhere. This is our planet. We need to characterize the diversity of the microbes on this planet, even if it seems intractable. So we also need what I would call niche information, which is not where something is found, but what the local environment in where you find different microbes. And this, again, lots of people are working on this. A lot of field microbial ecologists have been doing this for a long time. In terms of the Earth Microbiome Project, this relates a lot to the metadata that people would collect about particular samples, trying to collect information about the pH, about the salinity, about the local, density of the soil, the depth, the time, the temperature, etc. That information is going to tell us the niche of the organisms that we find, and if we get enough samples, we can start to look for patterns of particular taxa at any level. I don't know the offensive terms for the taxonomic hierarchy, not being British. Yeah, but we can talk about it later. But at any taxonomic level, we can start to ask questions about whether or not there's niche conservation in those particular taxonomic groups. So what I want to talk about for a couple of minutes is this issue of the field guide really is a representation of the biology of those organisms. So if you have a good field guide or a companion to the field guide, it tells you things like the behavior of a particular type of organism, whether or not it's going to hover, whether or not it's going to be seen running along the ground, what its sort of functional biology is. And we need that type of information for microbes, and one of the ways we have been going about trying to get this and to try and help inform metagenomic studies of organisms is to go back to those described taxa, the 6,000 to 8,000 described bacterial and archaeal species for which someone has been able to grow them in the laboratory, may have done an experiment or even hundreds of experiments on those organisms. And unfortunately, many of them did not have their genome sequenced. So I've been sort of obsessed for many years with filling in the phylogenetic diversity of life with genome data. And when I moved from Tiger, where I used to be, the Institute for Genomic Research, to UC Davis, I went to a meeting at the DOE Joint Genome Institute, and the director handed me an email, I think it was an email from Rick, that said something to the effect of, probably not offensively, for God's sake, we have to sequence all the described organisms on the planet. What is wrong with everyone? And Eddie said, what do you think of this? And I said, oh, that's great. We should just start with the phylogenetically novel ones. And we sent messages back and forth, and Rick and Folkermeyer were like, well, duh, of course we should start with the phylogenetically novel ones. And that's what ended up happening. So we started this project called the Genomic Encyclopedia of Bacteria and Archaea at the DOE Joint Genome Institute that has been marching its way through the phylogenetic diversity of bacteria and archaea, picking branches for which there are not available genomes, but for which there are cultured species, especially described type species, and sequencing their genomes, and filling in the phylogenetic diversity of life, giving us an enormous amount of functional predictions about different organisms, as well as anchors for interpreting metagenomic data about those taxonomic groups. And this project has continued. We've done about, I think, 400 or so genomes now as part of this project. All the data is being released for anybody to use, and if you want to go grab the data and publish a paper on it before we do, that's great. So having this onslaught of genome data and metagenome data and ribosomal RNA data, as I mentioned before, especially having good phylogenetic sampling of genomes, we've also tried to design new tools to help analyze the data. So we've taken that Amphora program, and Stephen Kemble, a postdoc in Jessica Green's lab at Oregon, has worked on combining all of the different genes that we were looking at separately into a single analysis. So you can scan through metagenomic data and try and anchor every single piece of a phylogenetic marker gene like ribosomal proteins and ribosomal RNA and REC-A onto a single phylogenetic tree to try and look at the phylogenetic diversity in the sample. And again, in part based upon stuff Rob has done, trying to leverage phylogenetic diversity as opposed to just taxonomic diversity of these environmental samples. This is a picture of the phylogeny of 31 marker genes relative to some metagenomic data. And again, you can scan through massive data sets to do this. What we've been doing with this genomic encyclopedia data is trying to build new marker genes. So if you focus on a particular taxonomic group like cyanobacteria, you might be able to identify good phylogenetic marker genes in that group there are maybe more of them than work for all bacteria. And so we've been scanning through all of this genomic encyclopedia data with an automated method I can tell people about and finding as many as we can phylogenetic marker genes for particular clades. And so, for example, we found 455 new phylogenetic marker genes for the epsilon proteobacteria, 286 for the Bacteroidetes, 106 broadly for the Archaea. So these markers, we have hidden Markov models for all of them, and people can use them to scan through metagenomic data and classify it in some way. We've been, people in my lab have been developing what we were originally calling Amphora2, and it's now called PhyloSift. And we're trying to use a gold mining motif, being near the 49ers Sacramento gold mining area. And Aaron Darling, Holly Bick, Guillaume Jospin in my lab, and Danying Wu, as well as Eric Mattson at the Hutch, have been working in part on this. The software is available on GitHub for people if you want to look through it and play with the currently available software and look at the notes. We're working on developing markers now for viruses and eukaryotes to try and cover the phylogenetic diversity of organisms. And again, part of the point of this is to scan through all of this metagenomic data and assign things to some type of taxonomic hierarchy from finding phylogenetic marker genes. Now, the reference data that we're using for all of this comes from cultured organisms. Most of the branches in the tree of life, especially the bacterial and archaeal branches of the tree of life, don't include cultured organisms. So this is a problem. There's now a spin-off project at the DOE Joint Genome Institute that's called the GIBA Genomic Encyclopedia Uncultured. Tanya Wojcicki is leading this and scanning through environments or particular samples using methods like optical tweezers and flow sorting and other approaches to try and get isolated single cells from a particular taxonomic group, and then use whole genome amplification or other methods to sequence the genome from those representatives, and now build up a catalog of the full diversity of life with reference genome data. So I don't know how much time.
5 minutes into the session.
Yeah, great. Okay, so the last thing I want to talk about is this issue, which I think is part of why I really like the notion of a field guide as opposed to, say, monographs, which is this engagement of the amateur community in making use of the information. And with birds, this is amazing. So there are an enormous number of citizen science projects involving birds. The Cornell Bird Observatory has upwards of 100,000 people involved in going out and surveying birds from the Christmas bird count and a variety of other approaches to gather information, making use of citizen scientists to help build feedback into the field guides and revise the information that you have there. We organized a workshop 2 weeks ago at UC Davis on citizen microbiology. We tried to find all the citizen microbiology projects that we could find in the country and around the world. We found 6. There probably are more, and we'd love to hear more. We had a discussion of some of the challenges and complications with citizen microbiology, such as for many of the microbes, you can't see them, which is a little bit of a challenge. But how to engage people in interest in microbiology and have them feed back into the studies of the microbial diversity and things like the Earth Microbiome Project. There is actually enormous potential here. People really are interested in the microbial diversity on the planet if you give them a way that they can participate in studying it and visualizing it and engaging in it. So you can go to the MicrobeNet is a website that we maintain for this Sloan Foundation project on microbiology, the built environment, and we have information about the citizen microbiology projects there. And the last thing I want to mention, which has already been hinted at, is to really do this well, we need to be open about many of the things that we do. We need open publications, we need open data, open tools, and open discussion. There are many examples of this from the microbiology world. I would like to put one plug-in for, I think, something that Rob is involved in, although I'm not 100% sure what this is. The Open Microbiome Initiative is an effort to really do this for microbiome projects. And one thing that I would like to point out is some of the sequence alignments that people use to classify organisms from their ribosomal RNA, such as those from the Ribosomal Database Project and from Silva, are copyrighted. And technically, you are not allowed to reshare those sequence alignments after you use them. And I find that unfortunate and not very open. And we need the development of open, completely shareable resources for this community in order to catalyze the spread of analysis and the spread of information. So I'm just going to end by saying I think there's lots of things people have done in plant and animal studies in the field. There are actually lots of things that microbiologists do in the field. And what I'd like to bring to that, and I think is actually possible, is this field guide to the microbes to help link the field studies that are going on to this sort of broader concept of a field guide. And I'll just leave it up there with my acknowledgments, and like the others have said, there's an enormous number of people involved in many of the things I've talked about, and I'd like to get everyone else, the entire community, involved. Thanks. Yeah.
For the viruses, have you looked at genes like G23 or G20 for those type of studies?
Yeah, so what we've been doing with viruses is similar to the approach that we've been doing with other genomes, is we took a database of viral genomes and we've searched them against each other to try and build gene families from that, and then flag the gene families that are more universal than others. I don't know what we just did this last week. I don't know which gene families are in this list. Other people have done similar approaches. It needs to be updated sort of constantly, especially with viruses, because we're so early in even getting genome data from the broad diversity of viruses in order to then generate these data sets. But what we do with these families is we ask, how well does an individual gene recapitulate the information in the entire genome? So that tells us whether or not an individual gene is in essence a good phylogenetic marker. And that's an important part to do, because you can analyze all genes. You can do a blast search with all genes, but that doesn't mean that each gene is actually useful. Yeah?
So Jonathan, I'm a big fan of citizen science and this open movement, but what we need are stories like Galaxy Zoo and so on of the real achievements that participants have made. And I'm curious if you have a couple of anecdotes about real achievements of amateurs.
So the best achievements in amateurs in this area are for the macrofungi. So whether or not you want to call mushrooms microbes would slide into this debate. But there was a great discussion of this sort of citizen microbiology project that Tom Bruns runs through Berkeley, where they have people going out and collecting macrofungi from Point Reyes and Yosemite and a variety of other places, taking pictures, recording GPS coordinates and the local environment, sending in those samples, and they've been doing traditional classification of them as well as DNA barcoding type of DNA-based classification, and they have increased the diversity of what's known at Point Reyes by 300% or something to that effect. And they're just starting in Yosemite. They have hundreds of people doing this. So the total phylogenetic diversity that they've been able to cover and ecological diversity has been immense. The other projects are earlier on. There's a thermophiles in your water heater project, which is really cool. There's a nitrogen metabolizers in your fish tanks, which is also great. You can go to our website and see these. There's Rob Dunn and Holly Menninger and people at the Wildlife in Your Homes sort of project have been starting to do microbes associated with those studies. This guy, Jack Gilbert, you may have heard of, is starting to do some things related to microbes in people's houses, etc. And I mean, many of them are very early on. The mycology, the macrofungi one has got the best results so far. So we don't have stories like the Galaxy Zoo, and that is clearly what we need in order to convince more people to do it. Yeah?
When you speak of a field guide, it strikes me to think of these problems. It's an amazing session as a naturalist. And what I'm struck by is that to date, you say 98 to 99% of the microbes cannot be cultured. Do you think that is a technical issue, or do you think that is actually telling us something important about microbes within the broader community metabolism?
Yeah, I mean, I personally think that in most cases, people have not thrown the book of potential culturing methods at most systems. What people a lot of times do is they know how to do one type of culturing, and they go to different environments and they apply that type of culturing to those different environments. There have been very few cases where people have said, let's go to this environment and throw everything at it. everything for years and years and years. And there are great examples, Steve Giovannoni with some of the open ocean bacteria that were only grown by massive serial dilution because they don't like to grow in large densities. Tom Schmidt has started a project at Michigan State to try and culture the organisms in the human microbiome and has cultured a huge number of ones that were never cultured previously by throwing diverse culturing methods at them. So I think a component of it is people haven't tried everything. I think another component of it probably is that some organisms have complicated interactions with other organisms, where they're sharing metabolites or structures, they have physical structures to their growth, chemical signals, etc. And we probably can reproduce those eventually in the laboratory, many of them. But they're going to be much more complicated to try and reproduce. You know, like people grow various pathogens in artificial tissue culture or things like that. I think that's what's going to be needed. So I think it's unclear whether or not how complicated some of these growth requirements will be. Yeah.
And to follow up with a small comment, I think there's two things I didn't see today. One of them is I saw lots of networks, but I didn't see any holarchy. I didn't see any expression of nesting, and that these microbes are nesting. The second word I didn't hear today was biofilm. And what's striking me from the work that I'm doing and what I'm hearing today, this is unbelievable what I'm hearing today, is that I think that what this project is doing is it's propelling us to a revision of or really a complete review of what life actually is, and seeing life not simply as a compartmentalized metabolic process, but that it's actually it's a biochemical consortium, and that cellular or microbial life is actually a member of that family. And I'm so interested in learning more by the great work you guys are doing. So just cheering you on.
Thanks. Any more? Yeah.
It seems to me that this is a really great opportunity for a number of educational schools. I mean, I like what you're saying here about involving amateurs in the process. A lot of times, undergraduates might qualify as amateurs in this field who we want to develop into professionals in this field. And I'm just curious if there are endeavors underway to try to make those distinctive and specific collaborations with predominantly undergraduate institutions or undergraduates in their own institutions.
Yeah, I mean, there have been many efforts in this area, and there are some that are ongoing. A great example of this is something Malcolm Campbell from Davidson College runs, this Genomics Consortium for undergraduates. I can't remember the acronym exactly, but he's been... GCAT. Oh, yeah, duh. Is the A for arsenic? So there are many examples of this where people have tried to do this. One that I know of a lot is the DOE Joint Genome Institute has an educational program run by Cheryl Kerfeld, who used to be at UCLA and ran an undergraduate genomics program there. And as part of this education program at the DOE GGI, she runs courses to train instructors in how to have their students get involved in annotating and analyzing microbial genomes. And we've had an Adopt a Microbe program for the genomes that we're sequencing with this encyclopedia, and various institutions have been adopting those organisms to try and either help annotate them or grow them and actually study their biology. Actually, Malcolm Campbell again they actually published a really cool paper on this with a couple of the genomes that we had sequenced. So there are efforts in the genomics world to do this. There are some in the ribosomal RNA survey world that are trying to both teach it at the undergraduate level as well as engage them in the actual science and surveys, etc., of these projects. So there are quite a few examples of this. It's just sort of gotten cheap enough to do in the context of, like at Davis, I teach a 700-person class. We have 2 microbial labs. This is biodiversity class. We have 2 microbial-focused labs in that class. We're going to change one of those labs to do more of this type of work. But we wouldn't have been able to do that 3 years ago. Okay. I can stay here?
Yeah. And So we still have a little time left in this session, and I don't know if any of you still have energy to discuss more. So what I'd like to do is ask the speakers to come up and maybe grab a couple chairs. You can, or stand. And we can have 20 minutes for general discussion. Did you have a question?
I have a general question.